Our next session is a keynote session presented by Professor David Anderson from California Institute of Technology. Professor Anderson, if you'd like to join me onto the stage, please. And the theme of your presentation is the neural circuitry of sex and violence. Thank you.
Thank you. It's a pleasure to be here. I'd like to thank the organizers for the privilege of inviting me to participate in this exciting interdisciplinary conference. All of us make moment-to-moment decisions that are influenced by the sensory cues that we receive from our immediate surroundings and also by our internal states of motivation, arousal, and emotion, and how those states are encoded in the brain and how they interface with sensory cues to control decision-making is a fundamental and poorly understood problem in neuroscience. That's particularly important because, as you've heard in the previous fascinating session, disturbances in these internal states, such as emotion states, are strongly associated with psychiatric disorders. Now, as a biologist, I'm interested in using powerful tools for probing neural circuit function, about which you'll hear more in the next session, to try to understand the basics of how internal states and behavioral decision-making are controlled in model organisms. And we find it particularly useful to study that in the setting of what I would call survival behaviors, such as mating and aggression. These are evolutionarily ancient behaviors that have a strong heritable or innate component to them, and they're essential for the survival and propagation of most species. And what this this illustration describes so beautifully is the complex interplay between signals in the environment and internal state. So, viewed from the perspective of this lion, he has to make a decision between whether to engage in mating with this female or aggression with the male, and if to engage in aggression, to direct it towards the male and not to the female. Interestingly, although the male does not attack the female, the very presence of the female also promotes the state of aggressiveness in the male. And so it's this interplay of how these states are encoded, created, and interact with external sensory cues that we are trying to understand. Now, there's a long history of understanding how internal states of motivation or arousal or drive are encoded in the brain, and it's always helpful to remember that we stand on the shoulders of giants. One of them, Konrad Lorenz, the German ethologist, conceptualized drive states with a hydraulic metaphor, where he viewed the brain as containing some kind of a liquid, the height of which created a certain pressure that drove the animal to engage in particular behaviors, with different behaviors released by environmental cues according to the strength of the cues and the level of pressure in this vessel. Now, this was a metaphor. Lorentz did not propose how this might be instantiated in the brain, but the assumption has been that because of its inherently fluid nature, that these drive states would incorporate or be based on mechanisms that involve diffusible neuromodulators like hormones or biogenic amines or neuropeptides. There's been relatively little consideration of the extent to which such models, which are basically a type of leaky integrator model, could be encoded in neural circuit dynamics. Now, Lorentz's contemporary and co-Nobel laureate, Nico Tinbergen, the Dutch ethologist, also thought about how behavioral decisions were controlled. He viewed them as being controlled by a series of feed-forward, hierarchically organized neural circuits that would gradually partition behavior through various centers in the brain into progressively more specific outcomes. He viewed these internal states, such as hormones, as acting at relatively high nodes in such a hierarchy that would influence a panoply of behaviors. For example, he grouped fighting and mating within a so-called reproductive hierarchy. Again, this was a speculation on his part in whether this type of hierarchical feed-forward model is true, and if so, where the state influences act in such a circuit remains to be determined. Now, Tinbergen was strongly influenced in his thinking by another giant, Walter Hess, who received a Nobel Prize in 1943 for his landmark studies showing that an emotion response of so-called defensive rage could be evoked from a cat by electrical stimulation of the medial hypothalamus. And this was one of the first times that it was shown that electrical stimulation of the brain, that is DBS, could promote an entire emotion state. And I think you can immediately see the link here to Helen Mayberg's beautiful talk. Since that time, it's been a pressing question as to where exactly are the neurons that are responsible for controlling this aggressive behavior, and what controls their normal function, and do they play a role in normal aggression? Now, this approach of brain-stimulated aggression was carried forward over the next 7 decades into many organisms, including rats and hamsters and even non-human primates. And particular work in the Dutch school by Menno Crook and his colleagues performed progressively more precise electrical microstimulation experiments to try to map the location in the rat brain from which electrical stimulation would release aggressive behavior. And these experiments defined a so-called hypothalamic attack area, which is highlighted here in orange. And as you can see, it's a fairly broad area that overlaps part of the ventromedial hypothalamic nucleus. For those of you who work on the cortex here, this is the other 80% of the brain that you don't talk about, which is down in the basement of the brain. The cortex is up there. Nevertheless, because of the microstimulation techniques that are used, which activate fibers of passage, it was not possible from this type of study to delineate the identity and function of the neurons responsible for this behavior. So when we entered this field now, back in about 2006, 2007, what we wanted to do was to apply emerging genetically based tools for mapping, marking, and manipulating specific sets of neurons in order to try to deconstruct aggression circuitry. And we felt that the best system to do this in a mammal would be in the laboratory mouse because of the genetic tools that are available in that system. However, in the 70 years since Hess reported his original observation, in cats there was no published report of brain-stimulated aggression with microelectrodes in the mouse, and that's because the experiment doesn't work. And I'm happy to discuss the reasons for that later on. And so we decided to take a genetic approach first to try to identify genes that would mark neurons in the brain that are associated or active during aggressive behaviors, and then to use genetic tools such as optogenetics to ask whether these neurons play a causal role in aggressive behavior, both with respect to their sufficiency and also, importantly, their necessity. So we focused on the ventromedial hypothalamic nucleus, which is about 6.5 millimeters below the surface of the brain. And we use the Allen Brain Gene Expression Atlas to scan for genes whose expression marked subpopulations of neurons within this structure. And our attention in particular was drawn to the expression of the type 1 estrogen receptor, also known as estrogen receptor alpha, whose expression beautifully is encompassed by the ventrolateral subdivision of the ventromedial hypothalamic nucleus. This is a region in which cells are activated during social behaviors like aggression and mating. And indeed, we confirmed by performing double labeling for expression of the estrogen receptor in red and CFOS, a marker of neuronal activity, which is in green, that many of the estrogen receptor positive cells are activated during episodes of fighting and to a lesser extent mating. So what we next wanted to do was to move from this look but don't touch type of experiment to one where we could actually functionally manipulate these neurons. And to do this, we had to use molecular genetic technology to engineer transgenic mice in which we could specifically manipulate genetically the estrogen receptor neurons in this part of the hypothalamus. And we did that by using embryonic stem cell technology to generate mice which had a genetic tag in estrogen receptor expressing neurons, in particular that expressed an enzyme called Cre recombinase, which you can think of as a kind of molecular key that can open a certain kind of lock. And what that allowed us to do, in fact, was to inject into the hypothalamus of these mice a virus that contains a locked version of a gene that you'll hear more about this afternoon called channel rhodopsin, which is a light-sensitive ion channel from the blue-green algae Chlamydomonas. And that channel, when expressed in any type of neuron, as shown by Ed Boyden, Carl Diceroth, and others, is capable of activating those neurons. So one can control or induce electrical activity with light in these genetically modified neurons. Now, because this virus is locked, the only cells that will express this ion channel are the ones that express this genetically engineered key. And so what one can then do is to thread a fiber optic cable into this region of the brain, connect it to a laser, and then specifically stimulate within that region of the hypothalamus the neurons that express the type 1 estrogen receptor. And when you do that, you see a result such as that shown in this movie. You'll see when the light comes on, the white mouse is a female mouse, which is normally never attacked by a male. And so here you see that stimulating these neurons causes attack towards a female. And when the light is turned off, the attack immediately stops. Now, this isn't because the stimulation is making the male mouse think that the white mouse is a male instead of a female, because we can stimulate these neurons and even trigger attack against an inanimate object, in this case an inflated rubber glove. And recent work from the lab of my former postdoc, Dayu Lin, who is now at NYU Medical School, has shown that activating these neurons can actually promote an operant conditioning paradigm where the mouse pushes a bar to get access to another mouse that he can beat up. And so that suggests that these neurons, yes, mice will work to get access to someone they can beat up. These neurons may control or contribute to a state of aggressive motivation, at least by that operational definition. Now, are these neurons actually required for normal aggressive behavior? This was a criticism that was leveled against Hess's early experiments. They were all gain-of-function experiments. And with optogenetic experiments, we can shut off neurons as well as turn neurons on by expressing a different light-sensitive ion channel, halorhodopsin, which is a chloride pump. And so here, when the light comes on, what we're doing is suppressing the activity of these estrogen receptor neurons. So in this movie, you'll see a naturally occurring fight beginning between the resident male in the cage, who has the fiber optic cable, and intruder male, and then you'll see what happens when the light comes on. So you can see that inactivating these neurons can stop an ongoing fight in the middle of its progression, indicating that ongoing activity in these neurons is necessary for the performance of attack behavior. Now, we can time precisely when we inactivate these neurons, and we also found that inactivating these neurons at the moment of approach can stop the resident from approaching and even beginning to attack the intruder. So these neurons are required at both the appetitive or the investigative stage of a social interaction and also in its consumatory stage, which is attack. Now, what's the relationship between these neurons that are necessary and sufficient for attack and those that control mating behavior? So it's been postulated by many investigators, including Sarah Newman, that male aggression and male sexual behavior utilize a similar network of subcortical brain regions, but that the level of activity differs in these regions according to which behavior is being performed. And this is, of course, all based on between-subject comparisons and post-mortem analyses of activity, and therefore it's not possible to determine from this whether, if this model is true, whether this means that the same regions and the same neurons are involved in both behaviors, or the same regions but different neurons within these regions are involved in both behaviors. And this became relevant to us because as we adjusted precisely the conditions of optogenetic stimulation of these attack neurons in mice, we received a surprise. And that is that low intensity of stimulation, rather than producing attack to an intruder, would promote investigation and, as you can see here, mounting behavior. And that mounting behavior would be exhibited irrespective of the sex of the intruder, whether the intruder was male or female or a castrated male. And in this case, simply increasing the intensity of activating the same neurons in the same mouse could switch the behavior from this sniffing and mounting behavior to attack behavior. So that was a striking observation, because it suggested to us that the neurons, and we're talking here about 1,600 to 2,000 neurons on each side of the brain, out of 100 million neurons in the total mouse brain. So we're dealing with a few thousand neurons, the activation of which can control different phases of a social encounter, investigation and attack, and different types of social behavior, mounting or aggression, depending on the level of activity in this population, at least the level of artificially induced activity. Now, what's the cellular mechanism to explain this phenotype? In some sense, this is a potentially artifactual phenotype because we're artificially manipulating the neurons, but by understanding the cellular mechanism underlying the phenotype, we may gain insight into the natural circuit-level processes that control these behaviors. One can think of two general classes of models to explain this scalable threshold-dependent control. In one model, the type of behavior that the animal engages in would simply be determined by the number of active neurons or the level of activity within these neurons, and that level of activity would be sensed by some downstream circuit which had a lower threshold for investigation and mounting, and another downstream target which had a higher threshold for attack, with some negative feedback thrown in to produce a switch. And this would be one example of a neural instantiation of Lorentz's hydraulic control of behavior model, albeit at the level of circuit activity. Alternatively, just because we've used a marker, estrogen receptor, to manipulate these neurons doesn't mean that those cells are all the same just because they express the estrogen receptor. So there could be different subsets of neurons within the estrogen receptor population, some of which are specific for attack, and those might have a high threshold for activation, and other of those neurons might control mounting or close investigation, and they might have a lower threshold for activation, for example, according to the level of tonic inhibition on these cells. So how to distinguish between these alternatives, and so we've done some electrophysiology, but most recently we've incorporated, in collaboration with Mark Schnitzer, who you'll hear from this afternoon, revolutionary technology developed in his laboratory, which allows one to actually record in real time the activity of a genetically marked population of neurons deep in the brain during a social encounter in freely behaving animals. And so what we have here is a male mouse with one of Mark's miniature microscopes. These are now known throughout the field as Schnitzer scopes, although I'm sure Mark will not call them Schnitzer scopes. But the mouse has a Schnitzer scope, and you'll see an intruder. And up here in this square, you'll see the field of view deep inside the brain of these estrogen receptor neurons, which have been genetically engineered to express a fluorescent protein that gets brighter as the neurons get more active. So the neurons literally glow brighter and flash as they become more active. So we introduce the intruder, and now suddenly you see activity appearing in this region, even as the resident is attacking and interacting with the intruder. And this was already a big step for us because we were concerned that the animal wouldn't engage in mating or fighting with this large microscope on its head. Now, one of the things that makes this type of research particularly challenging to apply in freely behaving as opposed to head-fixed animals is that the animals are deciding what behavior they are going to engage in at any given moment, not the experimenter. And so it becomes critically important to be able to quantify objectively objectively and reproducibly the behavior of the animal during these dynamic social encounters at a frequency of 30 hertz, which is the frequency with which we are imaging the neurons. And so we've invested in collaboration with Pietro Perona, a professor of machine vision at Caltech. We've invested a great deal of effort into developing systems for automated monitoring and classification of social behavior in pairs of interacting mice. And so we use a combination of video cameras and also depth cameras, which detect the depth of an object using reflected infrared light, sort of like the Wii games that you may play. And what we can do is collect that data and then train a classifier based on ground truth analysis of the videos to predict with a high degree of accuracy whether, for example, the males are engaged in attack behavior or sniffing behavior, or in the case of females, whether they are engaged in mounting behavior or investigative behavior. So this for us is a huge breakthrough because it will greatly increase the throughput with which we can correlate behavioral measurements with analysis of neuronal activity, and will increase the objectivity as well. And I think that there's a growing movement. I know that there's a growing body of work in the field more broadly to apply these approaches to better quantify and understand animal behavior, which hopefully will have some impact down the road on the pharmaceutical industry in the screening of drugs for the treatment of behavioral disorders. So we hope that that will help us in going forward, both in better understanding the functions carried out by the neurons within this tiny nucleus, and also to understand how those functions are read out by interconnected parts of the brain. And so as you can see from this diagram, VMH is simply one node in a larger interconnected network that conveys sensory input from the olfactory system deep into the brain, and that relays then output to the spinal cord. And the hope is that by performing analyses of behavioral activity and manipulating the function of these neurons in different regions of the brain, and in particular, we hope, once Mark develops the technology, to be able to record from multiple areas simultaneously in a deep brain region in a freely behaving animal, we will gain further insights into how this seemingly simple, but nevertheless clearly very complicated behavior is controlled. We may even someday get to the question of whether this other part of the brain, which seems to be completely dispensable for these behaviors, I forgot to mention, may play some role in regulating those behaviors in the sort of top-down manner that it is widely advertised to do, at least in humans. So the take-home messages from this portion of the talk is that we've identified hypothalamic attack neurons. They express the type 1 estrogen receptor, intermingled neurons in a tiny brain region far below the level of resolution of any fMRI experiment, control orthogonal behaviors, attack and mounting. These neurons may transform sensory signals into a representation of sex and internal state, and that this intensity of the state and different thresholds may link these states to behavioral decisions. Now, is there any possible translational relevance to this? Some of you may be thinking, why are male attack neurons expressing the estrogen receptor? I thought estrogen controlled female behaviors and testosterone controls male behaviors, and that is indeed the popular view. However, a great deal of work from Balthazar, Bell, Marler, Shah, and others has indicated that only part of the effects of testosterone on male-specific aggression are directly exerted through the androgen receptor. Many of the effects of testosterone on aggression or sexual behaviors are mediated by its conversion to estrogen by an enzyme called aromatase that catalyzes a one-step reaction. And it's the action of estrogen in the brain that is critical to the functioning of these neurons. And the regions of the brain that I've been telling you about are full of neurons that express the enzyme aromatase. Moreover, male mice with a knockout in the aromatase gene show little or no aggressive behavior, and treatment of mice and other animals with a drug called fadrazole that inhibits aromatase has a strongly suppressive effect on aggressive behavior. Interestingly, aromatase behaviors have been used in humans for decades as adjuvant chemotherapy for breast cancer in women, but have never been tested in human males for their ability to suppress irritability, anger, or aggressiveness. And we've been trying hard to interest some of the pharmaceutical companies in the US in an open-label trial for this, but none of them is interested because these drugs are off patent and available for pennies, and so there's no money to be made. Another great example of how potentially useful discoveries are limited by economic incentives, at least in our country. Now, I'd like to just finish briefly by telling you something about our work using Drosophila fruit flies as a model for studying aggressive behavior. I love this quote from Charles Darwin: "Even insects express anger, terror, jealousy, and love by their stridulation." My colleague Seymour Benzer at Caltech opened up the use of Drosophila as a model for studying behavior to the genetics community in the 1960s. For those of you who have never seen fruit flies fight, you haven't been looking at your bananas closely enough. You see this snapping motion where one animal snaps down on the other. It doesn't look like much at 30 frames per second, but in this movie from Martin Heisenberg at 5,000 frames per second, you can see what a lunge looks like, and there are some spectacular but rare examples that Heisenberg's group captured as well. So we're not supposed to anthropomorphize our organisms, but if this is not suggestive of malevolent intent in flies, I don't know what else I could show you that might be. Now, we can't stick electrodes into a fly's brain or even fiber optic cables because it's too small. So we can take apart flies' brains genetically, and we can do that because there are libraries of thousands of different strains of fruit flies, in each of which a specific subpopulation of neurons is reproducibly genetically labeled. And we can use that to activate these neurons either optogenetically or thermogenetically and see what behavior those neurons control. And so, for example, if we express a warmth-sensitive ion channel which opens up at 27 degrees, we can put this ion channel in each of these different lines into these different neurons, turn the neurons on, and in an unbiased way ask, do any of these neurons increase aggressiveness? And again, as in the case of mice, here the development of automated methods for tracking and measuring behavior has been critical for carrying out this type of high-throughput, forward genetically based analysis. So we decided to carry out an initial pilot screen for neurons that express neuropeptides that might be involved in aggression. You've already heard about neuropeptides like oxytocin and vasopressin from Dr. Meyer-Lindenberg, and it's clear that neuropeptides play an important role in controlling basic survival behaviors. And so we asked the question, are there any neuropeptides that are involved in controlling aggression in Drosophila? And if so, which ones are they? And we did that by making a library of flies in each of which we could activate at will a different subpopulation of neuropeptide-containing neurons. And when we did that, we found that flies in which we activated neurons expressing a neuropeptide called tachykinin showed an extremely high level of aggression. Now, because these neurons are genetically labeled, we can look inside the brain and we can see, oh, there's a group of about 50 to 100 neurons that are labeled in this brain. How do we figure out which of those neurons are controlling aggression? Here we took advantage of the fact that in flies, as in most other species, including us, males are more aggressive than females, and we compared these neurons in male versus female brains, and we found that one cluster of about 3 to 5 neurons was detectable in the male, but not in females from this same genetically marked strain. Now, work from a number of laboratories, including Barry Dixon, Bruce Baker, and Daisuke Yamamoto and others, have shown that male-specific neurons involved in courtship express a master gene called fruitless, and we confirmed that these neurons express the fruitless gene. And so in flies, we could carry out genetic intersection techniques and label only these neurons, which are male-specific and fruitless positive. And when we did that, we found that activating those 3 to 5 cells increased aggression, and inhibiting those 3 to 5 cells blocked aggression. Moreover, there was no effect on courtship. So these are male-specific neurons that specifically control aggression. And finally, we were able to replicate a version of Nico Tinbergen's famous dummy experiments, showing that we could, by overexpressing this peptide in these neurons in the fly, get it to attack a dummy, a moving fly-sized magnet. Interestingly, tachykinin has been implicated in aggression in mice and in rats by genetics and pharmacology, and moreover has recently been shown to be expressed in the estrogen receptor ventromedial hypothalamic neurons that I told you about are important in aggression. And there are correlative studies suggesting that there is a correlation between levels of substance P, which is a tachykinin family member, and self-reported levels of aggression in the cerebrospinal fluid of patients with borderline personality disorder. So simply what I want to say by this point is that comparative studies are key, both in vertebrates and invertebrates, in understanding and revealing general principles of brain function at the cellular and molecular level. And I just want to emphasize, end by emphasizing the fact that violence is a symptom domain of many psychiatric disorders. That does not mean that all individuals with psychiatric disorders are violent. That's Logic 101. But many people with violent behavior may have an underlying psychiatric disorder, particularly in the area in the United States of gun violence and sexual assault. We have 80,000 prisoners in solitary confinement in the United States for violent crime, costing the country approximately $6 billion a year. And yet the National Institute of Mental Health does not consider aggression and violence one of the main topics of relevance to its research mission. And with that, I will thank the talented students and postdocs in my lab who've worked on mouse aggression and fly aggression, and our collaborators at the Allen Institute, Stanford, and inside Caltech. And thank you for your attention, and be happy to take any questions.
Thank you. No, please stay, because you're going to go and sit down. I'm sorry I've slightly encroached on you, but we've nearly missed our tea break. But that was truly, truly fascinating. So why don't you take a seat? Right. Thank you ever so much. I'm now going to ask Professor Isabelle Mansuy, who works in neuroepigenetics at the Medical Faculty of the University of Zurich at ETH Zurich, who's going to lead a very, very short discussion. And I suggest that you don't ask any questions if you want a cup of tea. Thank you.
Yes, indeed, I have to be very selective in my questions. But first, I would like to thank you for putting it so, showing so brilliantly how different tools, a combination of cellular, optogenetic, and behavioral tools can be used to dissect out a circuit in relation to behavior. So the only question maybe I will ask, I will try to resonate with the previous session, and which relates to the question of nature versus nurture. So this type of not simple but ancient behaviors, mating and fighting. So how do you think they are really embedded or naturally expressed, and what is the influence of the environment on these behaviors?
Thank you, Isabel. That's a fascinating and very important question that I didn't have a chance to touch on. This is a debate that's been raging in the academic community since the 40s between the American Psychology School, who felt that behaviors like aggression were learned, and the ethologists who felt that they were innate. The answer is clearly both. How genes and environment interact to control aggression and where they do in the brain, we don't yet know, but we hope that beginning to unravel the circuitry will show us some of the places to look for those effects. I can give you one example that we have of a gene by environment interaction and aggression. So there are dozens of different inbred mouse strains that differ in their levels of aggressiveness. And in addition, there's an environmental manipulation, social isolation, which greatly promotes aggression. What we find is that genetic differences in aggressiveness are completely suppressed by housing the animals in groups. Only when the animals are socially isolated do the gene influences in aggression become apparent. So that's a beautiful example of a gene by environment interaction. What the genes are and what the environmental factors are, how group housing suppresses aggression, we don't understand, but it's a very important question for the future.
Now, if we take another argument that was discussed previously about the urbanicity, then how do you see, can you put this in the context of evolution, the fact that societies are a lot more urban than they used to be, and how you think this influences these circuits, granting they are plastic? We don't know about the plasticity. If you could also comment on what you think about the...
Well, I can only speak on this as a lay person because I'm not a sociologist or a social psychologist, but I am an ex-New Yorker, and having survived 8 years in Manhattan, I can tell you that life in a city like New York is far more competitive than it is in sleepy Pasadena, and I'm sure that many of our evolutionarily ancient motivational control systems that in earlier times and in other places in the world predispose us to violence also control our ability to hail a taxi cab or to get the best job in a Fortune 500 corporation in the corporate jungles of Manhattan.
Right now. Can we please say thank you very much for a fantastic presentation and a very, very interesting discussion afterwards. Thank you.